First thing I want to do, we have a new allergist in the community. Jennifer, I'd like you to just come up here and show your face to people, introduce yourself, tell them a little about yourself.
Okay, so my name is Jen Regan. I'm new to the area, just moved here a couple weeks ago. I just finished fellowship at Northwestern in Chicago, so I did my MD PhD there, so I've been there for the last 15 or so years.
Wow.
So I'm really excited to be here, and I'm excited to meet everyone that I haven't met so far.
Where are you going to be working?
So I'm working in Renton, Redmond, and Issaquah.
With Nick?
Yeah.
Oh, cool.
All right, thank you. And Frank had one thing he wanted to share with us as a follow-up on yesterday's presentation before Thao goes ahead with her presentation.
Thanks, Len. There'll be a test on this later. So, partly because, unfortunately, we had technical difficulties when Dan gave his excellent little presentation, and I had made a comment because I went to a Regeneron meeting about dupilumab, and I just wanted to put this up there because I had incorrectly stated IL-33, and I actually meant IL-31. And you can see that highlighted here, that is actually the one that's downstream from interleukin-4. So blockade of the common IL-4, IL-13 receptor should indeed have an effect on scratch. I had mentioned IL-33, which, as Teal mentioned, is actually upstream of that. One interesting thing that I came across is we don't have anything for humans, but there's a company called Zoetis, which many of you may have heard of. They actually make biologics for animals. And as the name implies here, this is one of them. It's called Vocavetnab. And it actually specifically targets IL-33.
31.
31, I did it again. It's, interestingly enough, it also, just like we call omalizumab a humanized monoclonal, it's called a canonized monoclonal. So it's actually made in mice and then remade to look like a canine protein. In case you're wondering if this is a popular item, this company last year had $6 billion in revenue. And for those of you who are worried about your portfolio, there's the stock ticker. A very interesting company out of Kalamazoo. All right, I just wanted to share the pathway.
Well, yeah, Frank, while you're up there, this is interesting. It reminds me back when John Milner used to join us. Those who are too young won't remember John was a dermatologist who used to give us the most interesting presentation. And he would always point out that antihistamines didn't work for the itch of atopic dermatitis, and it always made you wonder what caused the itch and the scratch. And I presume this is the answer, the IL-31?
Well, it's at least one of the major candidates and certainly proof of concept in these studies. Interestingly enough, the senior author on the papers on Locavetnab was actually Steve Durham.
Oh, yeah?
Yeah, so he's involved with dogs as well as people.
All right, well, this is today's topic.
Good morning, everyone. I'm Thao Tran, here to talk to you about alarmins. You know, driving through this area caused me to release a lot of alarmins, I think. So let's get started. I have a whole bunch of slides I'm hoping to get through. It's a very interesting topic. When Len gave this to me, I was like, I had no disclosure, by the way. I was like, where am I going to take this? Alarmins is a huge topic, out there. So I really want to focus on, reviewing and defining alarmins that has relationship to what we deal with, the Th2 pathway diseases, but I'll mention the world of alarmin in other diseases and their implications. But we'll define alarmins, we'll review some notable alarmins that's well studied in allergic pathways and the implications as biomarkers and as possible therapeutic targets. So alarmins. Alarmins are these quiet molecules that we've known and are endogenous to ourselves for many years now, but over the decades, their roles as dual molecules have been of interest. So they tend to be these homeostasis molecules in our nucleus and cytoplasm. But upon damage, upon injury, upon infection, they're released by these dying, damaged necrotic cells, and their roles change. The molecules may change a little bit, but their roles as pro-inflammatory cytokines starts at that time. And so one of the this is reviewing here. Alarmins act as immune modulating molecules. They're rapidly released into that damaged microenvironment. They are not released in programmed cell death in apoptotic cells. And once they're released as pro-inflammatory cytokines, they recruit both the innate and adaptive immune system. And their role is to repair, reconstruct that damaged tissue. But when that role is uncontrolled, then you have this kind of amplified inflammatory cascade in which we diagnose as diseases. So important alarmins in the airway. There are important roles in Th2 cytokine pathways development, and they're important in not just asthma, but in also atopic dermatitis, eosinophilic esophagitis, etc. But the ones that are well studied are interleukin 33, Thymic stromal lymphopoietin, or TSLP, and IL-25, also known as IL-17E. And their interests nowadays are becoming more popular because, especially IL-25, 33, TSLP, they're primarily seen and released in epithelial cells, keratinocytes, and their stromal cells. So these cells that are at the barrier of the outside and internal world. And damage at those sites causes their release into that area. And in that microenvironment, the pathway. Can I pull out is there a mouse? Oh, there it is.
Yeah.
This is the early signals of inflammation that can push both the immediate innate pathway response and eventually our known Th2 adaptive immune response, where we become antigen specific. And their roles are important because right now we have therapy focused on the later stages of cytokine release, but alarmins actually triggers the earlier stages. So modifications of this earlier stage may play a significant role in long-term therapeutic response, especially for recalcitrant and severe diseases. So interleukin 33, IL-33, is a member of the interleukin-1 family of cytokines. It's expressed primarily on epithelial cells along skin, eyes, respiratory, and intestinal tract. It's also expressed on endothelial cells, smooth muscle, and a lot of lymphoid organs and keratinocytes. And they're in homeostasis when they're within the nucleus. They play a very unknown role, but probably in chromatin compaction and private transcription repression. When the cells are damaged or an inflammatory signal is triggered, these or full-length interleukin 33 become processed, and then it's either passively released from those necrotic cells or actively released from an alternative pathway, rather than classic Golgi pathway. And upon the release and the protease activation, they become these small molecular weight smaller molecular weight form, they become very potent, actually very active cytokines for a whole slew of different pro-inflammatory pathways, as seen in both Th2 diseases and Th1 diseases. Their roles and their activity actually is controlled by their binding to the suppressor of tumorigenicity 2 receptor, otherwise known as IL-1 receptor, like 1 receptor. And the binding of IL-3 to these receptors activate the slew of pro-inflammatory response. Now, these receptors themselves are pretty ubiquitous. They're found on a number of inflammatory cells, but their prominence are known as the IL-33 can activate ST2 on basophil. This causes a significant increase in expression of IL-5, IL-9, enhances degranulation of the basophils, especially with IgE cross-linking occurring. They cause maturation of mast cells and the release of their cytokines and proteases. They activate eosinophils and also cause their degranulation. And more interestingly, you could see probably the most recent Jackie, the big talk on the innate lymphoid cell, and the IL-33 is known for their prominent activation of the innate lymphoid cell type 2. And this is an interesting activation pathway because innate lymphoid cell is probably one of the early cells that link the early innate Th2 response to eventual antigen-specific adaptive Th2 response. So these innate cells, as you can see, produces a slew of Th2 cytokines from IL-4 to IL-5 to IL-13. They play a role in, as a response to helminthic infection, protozoa infection also, but their role in actual control and homeostasis of injury is just mainly tissue repair and remodeling. But because of this potential pathway and the shift of pathway, it cannot push Th2 pathway too strongly and eventually causes allergic diseases. IL-33 also, to kind of control the Th2 pathway, they also suppress the regulation of T cells or Tregs. Initially, most of the earlier studies in IL-33, they saw a strong correlation with Th2 pathway diseases or inflammation, but actually recent studies in the past few years have shown that IL-33 is also expressed on a few important CD8 T cells and antigen-specific CD8 T cells, and they have these ST2 receptors. And in the setting of IL-12, they actually play a pretty important role in repairing tissue damage from various viruses like rhinovirus. So this concept being relatively new and being relatively recently studied, they're actually looking into the potential use of IL-33 as an adjuvant in vaccine, in CD8 positive T cell vaccines. So IL-33, its main regulation is through its attachment with ST2 receptor. When ST2 is expressed on these pro-inflammatory cells in this injury microenvironment milieu, there's also rapid and extensive release of what is SST2 or decoy receptor. They're a soluble receptor in that same microenvironment, and they actually, in the normal setting of injury, actually rapidly absorb a lot of these interleukin-33 cytokines, and consequently inactivate them. So that's the regulation and control behind it. I just want to review one of the major studies that actually associated IL-33 with adult onset asthma Asthma. This is Prefontaine and his group. They looked at IL-33 levels in bronchial tissue biopsy and bronchial alveolar lavage fluid and compared it between moderate and severe asthmatic with healthy control. And they looked at 4 mild, 4 moderate, 5 severe, atopic, non-smoking asthmatic with 5 healthy control. And this is just binding of IL-3 on the epithelial cell. In A is mild asthmatic, B is severe asthmatic. You see intense binding there, and C is actually a normal control. And D is just proof of concept that this staining works. They actually put in recombinant ST2 that binds the IL-33, and they're in the severe asthmatic biopsy, and they saw no staining. And so what's important is actually IL-33 intensity staining or presence in the epithelial cell is found significantly different between normal control versus the mild, moderate, and severe asthmatic, with a trend showing that IL-33 is expressed somewhat higher in severe asthmatic. And similarly, when they looked at the bronchial lavage, the IL-33 cytokine is higher in the severe asthmatic, moderate asthmatic, compared to the control. And when they looked at the presence of IL-33 mRNA in epithelial cells, it's found more higher in asthmatic than the normal control. So those are some of the, that's an example of a number of studies, early studies, that shows the association of interleukin-33 and is possible as a biomarker for asthmatic severity. This recent study in pediatric asthma, looking at severe asthmatic, was also very interesting. Again, one of their primary goals is to see if there's a relationship with IL-33 release in these models of severe asthma versus control. What was interesting in their data, so again, just an association with the presence of submucosal IL-33 in these patients. But what was interesting was this result. So this is reticular basement membrane and IL-33 present. The higher the collagen production or the thickening of the reticular basement membrane, the higher the presence of IL-33 in these patients as a trend. But more interestingly with this is a comparison of healthy control, asthmatic fibroblasts, those asthmatic fibroblasts with budesonide treatment, and then the treatment of those fibroblasts with budesonide and IL-33. As you can see, in normal control, collagen production is relatively low. In the presence of IL-33, collagen production is high, and when treated, asthmatic treated with budesonide, their fibroblast collagen production is reduced, showing concept of treatment. But when you actually add elevated levels of IL-33, collagen production is actually not as reduced when treated with budesonide. So the biggest question they have was, it suggests that IL-33 may possibly play a role in steroid resistance, in severe resistant asthma. So that area of study is, being looked into.
Just restarting the system.
Just hit reject.
And we had I'm sorry to interrupt you here. I got a text here. Are we having trouble broadcasting to this?
That's why it's being reset. So now it should be broadcasting. Travis is doing it on the outside. So we're good.
That was good for her to go ahead. All right, start over. We didn't understand it up till now anyway.
Pathway, and also disinhibit responses to other physical injuries such as mechanical injury, cardiac and ischemic diseases in brain and cardiac pathways. So the consequences of it is important to look at. So.
With that, since it plays a role in maybe antiviral activity, that would be particularly troublesome. Have they tried any models where, if they impaired it with the monoclonal against IL-33 with RSV infection of maybe an animal model, did that make it worse? Or?
Yes, it actually made it worse in murine model, and that's where the concern is with the pathway inhibition. So.
I know this isn't your topic, but have they looked at that in eosinophilic esophagitis to see if IL-33 is one of the mediators there?
I think so. TSLP, which we'll talk about next, has been the more strongly associated because actually single nucleotide polymorphism has a strong association with EOE. But IL-33, talking about itself, like I said, I could probably have several talks by searching through all the literature about IL-33, but it has a strong association when measured from various diseases, from EOE, anaphylaxis, idiopathic anaphylaxis actually, Crohn's disease, ulcerative colitis, atopic dermatitis. And IL-33 and atopic dermatitis is very interesting because it may actually play a prominent role on the severity of the atopic dermatitis response in the setting of staph infection. Because you have both the pathogen signal as well as the Th2 cytokine signal augmenting each other that way. But yeah, and it goes on forever, And then, we're not even talking about the murine models of how it increases certain cancers. So.
Can you tell us the kind of stimuli that cause the release of IL-33?
Yeah, and any of the stimuli, so infection, so ranging from viral, bacterial to helminthic infection, actual physical injury, and the common physical injury, so IL-33 is actually strongly studied in smoking COPD. So the smoking is the physical injury, the thickening of the fibroblasts is the COPD, but again, the measures of IL-33 in that injured microenvironment is much higher than the normal control or mild COPD pathway.
So it seems like it's regulated through the ST2 receptor. So what's the latest on polymorphisms of that receptor and asthma risk?
So I think there's a lot of studies in mouse models with polymorphism, but yeah, there are a lot of in all three molecules we're talking about IL-33, TSLP, the IL-25, and their concurrent receptor. There's actually a number of studies on single nucleotide polymorphism on both the interleukin cytokine as well as the receptor associated with numerous atopic disease. I don't remember all of them, but it's interesting. Like I think in IL-33, the SNP polymorphism is actually associated. The rare SNP is associated with protection from asthma. Well, majority of them seem to be associated with asthma. So. Shall we move on to TSLP? So this is I'll try to go through this relatively quickly because it's a very similar pathway. All three of these alarmins, we should call them, are released from the epithelial cell, and they play a very similar role, and they augment each other. They induce one, Th2 pathway to induce the expression of the other alarmin receptors in these cells. So they do, in a lot of studies, you'll see the enhancement of IL-33 with IL-25, the enhancement of IL-33 with TSLP. So they are potentiating pathway together. But their TSLP, member of the IL-2 cytokine, polymorphism of TSLP gene is associated with asthma, atopic dermatitis, food allergy, and like I mentioned, there's a strong association with the risks for eosinophilic esophagitis. They're commonly expressed in the lung, skin-derived epithelium, but are expressed in fibroblasts, endothelial cell, mast cells, macrophages, granulocytes, and dendritic cells. They, similarly to IL-33, they are released from these epithelial cells with mechanical injury, with protease activity. So when neutrophils and mast cells release their proteases from inflammatory signaling, can cause TSLP to be released from these cells. Allergens that contains proteases can cause release, of rhinovirus. So in the setting of infection, double-stranded DNA through the toll-like receptor can cause release of TSLP. And when the TSLP is released in this microenvironment, their receptor is the TSLP receptor or TSLPR, and it's a heterodimer receptor that's associated with IL-7 or alpha subunit. And again, TSLP activity is also ubiquitous just because TSLP receptors are expressed on many cells also, from dendritic cells to both CD4 and CD8 T cells, Treg cells, the B lymphocytes, mast cells, to the eosinophils, and also the airway, epithelium, and smooth muscle. So it's augmenting this pro-inflammatory localized response. And this just go through like when TSLP binds to myelocyte dendritic cell, they produce IL-8, eotaxin, OX40 ligand. So you can see the activation of naive T cells occurring at this time. So TSLP is purported to play a very important role in stimulating that early innate injury over to Th2 specific pathway to produce antigen response in that way. TSLP also binds to the innate lymphoid innate lymphoid cells type 2, again producing amplification of Th2 cytokine. The natural killer cells is induced to produce IL-4 and IL-13. Mast cells are induced to further release their cytokine and also activate. It does very similar things with basophils and eosinophils. And with eosinophils, TSLP definitely appear to play a role in delaying their apoptosis and causing a higher duration length of eosinophilic inflammation. And it's interesting because there's a number of articles on the role of TSLP in antigen non-specific Th2 diseases, such as vasopolyposis, chronic rhinosinusitis, severe non-allergic asthma with eosinophilic feature, acute as well as chronic eosinophilic pneumonia. The reason I wanted to bring TSLP up was this is a New England Journal of Medicine article with Gabriel and his group. This is a proof of concept article, and what they were looking at is an anti-TSLP monoclonal antibody, and binding to the TSLP will prevent its interaction with the receptors, and it seems to ameliorate responses to the allergen challenge in this model. So they did a double-blinded placebo control of 31 mild allergic asthmatic who were on no treatment whatsoever. I think they made sure that they even cleared a beta agonist prior to the study, and they were assigned randomly to receive the 3 monthly doses of this molecule AMG 157 at 700 milligrams per dose. And this is the study design. So you can see they measured spirometry. They measured the sputum cytokine levels before and after an allergen challenge before treatment. Then they had 3 treatment at day 1, day 29, and day 57. They did one allergen challenge with measurements of levels before and after, and then they also did a post-treatment allergen challenge at that time. And what they saw was definitely interesting. So this is pre-treatment, day minus 14, and for patients, so these are mild asthmatic after allergen challenge. But for patients who receive blue is the placebo and treatment, they're about the same. Their drop in FEV1 was the same. Day 42 after the two randomized randomized treatment, you could see that the drop in FEV1 in the treatment group was less than the placebo group. Yeah, and then at day 58, there is a difference. Yeah, also. I mean, at day 84, there's a difference in the treatment AMG 157 group and placebo also. And this is true in both the early phase response to the allergen challenge and between 0 and 2 hours, and also the late phase response between 3 and 7 hours. There's also a difference in the blood eosinophil level between the treatment group and the placebo group also. So you can see this is before treatment, and then this is after treatment 1, treatment 2, and treatment 3. There is a difference, a lessening of blood eosinophils in the AMG 157 group. And this is a look at the sputum eosinophils also. Before treatment, they were very similar. Yeah, after treatment, with the allergen challenge, you can see the placebo group had an elevation in the sputum eosinophil while the treatment group had attenuation. And this was true also at day 84. And then another marker, inflammation, Th2 inflammation with the fractional excretion of nitric oxide. Again, the treatment group had a reduction of compared to the placebo group. And so this molecule is anti-TSLP, may be a potential treatment patient with allergic asthma. Seems to attenuate bronchoconstriction in the early and late phase response, and a reduced index of airway inflammation from to sputum eosinophil during the duration of the study. They don't have any conclusion on once the therapy stops, if the duration persists.
Right, Thao. My brother.
And the final.
So question on
know
the
anything about the future of that molecule? Is that being pursued?
I'm curious about it, but I did not find a lot of article after the New England Journal of Medicine article. So, yeah.
I had a question on, because asthmatic, you've been a site for the catnip study, the sub-Q immunotherapy with cat, plus or minus that AMG 157.
Yeah.
Do you know when those results are coming out, or
Is that the molecule that we're using?
I think so.
Yeah, that's the one.
I don't know.
Yeah.
Because it's a theory of that if you could have a really potent induction of tolerance by having a early blocking the skewing of the Th2 response early at the same time you're doing the sub-Q immunotherapy, it could be a real potent approach. You can have a shorter period. And
Kind of a related question: did you come across anything using that in like inducing tolerance in food OIT, or maybe creating more permanent tolerance?
The study's ongoing. I don't think we're anywhere close to knowing the results.
do you know when that will complete enrollment? Or?
Steve would have to.
I think it's another 6 months or so.
Mary?
And the final alarm that I want to talk to about when it comes to the strength of the epithelial injury releases, IL-25. There's less data on this, but they're still in the now. But they're produced by airway epithelial cells, Th2 mast cells. They're expressed in most of the epithelial cell in the extranuclear compartment. And again, they are released with exposure to proteases. Again, whenever there is infection leading to inflammatory release of proteases or allergen proteases. They bind to another heterodimeric complex of IL-17 receptor A and IL-17 receptor B. And this results in the expression and release of Th2 cytokine, and it antagonizes Th17 cell differentiation. And so consequently, they play an important role in clearance of pathogens from the mucosal surface. Their role is due to binding to their receptors, the IL-17RA and RB complex receptor, which are found on antigen-presenting cells, smooth muscles, airway epithelium, fibroblasts, eosinophils, and natural killer T cells, to name a few. And IL-25 is also important in promoting the development and activation of, again, the innate lymphoid cell type 2. And so the concept of ILC2 is that they release IL-5 and IL-13 to act like Th2 cytokine pathway inflammation, but without antigen-specific triggers. And it causes eosinophilic infiltration and mucus production. So their big role in diseases, pathway diseases like eosinophilic esophagitis, which may not have specific food antigen response, is being looked at. So, and there's polymorphism of those receptors for IL-25 that is associated with a reduced risk for asthma. And yeah, as mentioned here, minor alleles of IL-17RA was associated with also a decreased risk of asthma and exacerbated respiratory diseases. In a study measuring IL-25 mRNA levels in blood, sputum, and BAL, they, again, very similar to the previous alarmist study, looked at asthma cohort, and they tend to be, the more severe the asthma, they tend to be IL-25 high versus a mild asthma, IL-24 low subgroup. And the IL-25 high asthmatic subgroup tend to be more atopic. They respond to, they make more serum IgE antigen. They tend to have higher IL-13 expression and eosinophilic activation. There is a molecule out there that is an anti-IL-17 receptor A monoclonal antibody. So they inhibit receptor binding at the receptor site. And they looked at it as a potential role for moderate severe asthma, but did not see any therapeutic response from that. And they've also looked at it with a lot of IL-25 positive diseases such as rheumatoid arthritis, Crohn's, and psoriasis. And it looks like it has some benefit in the psoriatic inflammatory pathway only. And finally, this is alarmin, which I want to mention because it's pretty interesting also. This high mobility group protein B1 alarmin is not just found on the epithelial pathway. These are found ubiquitously through multiple cell types throughout our body. There are these non-histone nuclear protein that regulates DNA transcription, but upon localized injury is when they get released, and upon release is when they promote the inflammatory response, causing a whole slew of systemic issues. And these are, you could see studies for HMGB1 in sepsis to SIRS, etc., to a lot of ischemic injury, stroke, ischemic injury, reperfusion injuries. but in to talk about them a little bit, they during apoptosis, their end tail changes, and that's when they get released. Once they're released, they stimulate endogenous cytokine release of TNF alpha, interleukin 1 beta, interferon gamma, and they also mediate local inflammation with binding to toll-like receptor 2, toll-like receptor 4, and these RAGE receptor for advanced glycation end products. Their control, their regulation, because once they're released in an enormous amount, but their control is also strongly controlled because there's common and in homeostasis there's a release of these endogenous secretory RAGE receptor. Again, they're these soluble extracellular receptor that binds to any release HMGB1 as decoy receptor, and they inhibit their function. So HMGB1, they play a role in cardiac regeneration, revascularization, wound healing from that kind of ischemic injury or physical injury. They're implicated in sepsis, reperfusion injury, transplant rejection. They play a role in tissue repair and steroid injury. They are a large marker as a late mediator in that whole cytokine storm in SIRS and sepsis. But in adult asthma and pediatric asthma, they may play actually a very interesting marker of disease severity. So in Watanabe's group, they measured sputum HMGB1 and RAGE level in newly diagnosed non-smoking asthmatic.
Can I interrupt you?
Yeah.
I have no idea what RAGE is. Maybe
So
everybody else does.
RAGE,
But,
RAGE is just one of the receptors found on the inflammatory, the responding cells, that can actually bind to HMGB1 once it's solubilized, and it induces the whole pathway, post-transcriptional pathway of cytokines.
so it's a receptor for this?
Yeah, RAGE is a receptor. So in this study, they looked at the sputum of 44 adult asthmatic compared to healthy controls. And as you can see, HMGB1 levels is much higher in the asthmatic patient than the normal control, and the expression of RAGE is also higher in the asthmatic patient. And they divided the asthmatic based on the GINA, diagnosis. But you see the normal control compared to mild, moderate, severe asthma, HMGB1 correlates with disease severity. What's interesting was actually in C, their expression of soluble RAGE. Remember, RAGE is actually the decoy receptor. It controls, decreases the activity of HMGB1. So the interesting thing is the presence of RAGE is high in mild asthmatic, but you can see it's actually relatively decreased in comparison. It's higher in asthmatic in general, but it's decreased in moderate and severe asthma. So it may be a dysfunction, a poor release of these decoy receptor that actually is participating in the resulting in the presentation of much more severe asthmatic presentation. So the question is, if you do treat with, if you have a molecule that acts like a decoy receptor, would these severe asthmatic start to look more like a mild asthmatic in the setting of inflammation and release of HMGB1? And this is also a presentation that, data showing that there's an association with neutrophilic release and severity of asthma with release of HMGB1. rate was higher in asthmatic normal control, but no significant difference between the different severity of asthma, suggesting that failure of this negative feedback loop is associated with asthma severity. And HMGB1 also plays a role in as a neutrophil chemoattractant, so may explain some of the more neutrophilic presentation in more moderate to severe asthma. And it's HMGB1 is also proven to be a sensitive biomarker in pediatric asthma. And their group evaluated the sputum of HMGB1 levels in children off therapy of asthma, and they looked at lung parameter, and 50 children was compared to 44 healthy controls. And again, this is looking at HMGB1 with controls right here and mild, moderate, severe asthma. They're all significantly different, and you can see an increase in HMGB1 expression in severe asthmatic through mild asthmatic. And what I thought was pretty interesting was this, because the correlation was relatively strong. But yeah, this is the change in FEV percentage to HMGB1, and so this is sputum presentation of HMGB1, and you can see in A is mild asthmatic, the correlation. A is mild asthmatic, B is moderate, and C is severe asthma, and there's a strong correlation with the presence of HMGB1 in asthma. So this HMGB1 then consequently may play as a sensitive biomarker for allergic asthma in both children and adults, and it may be a good biomarker as it's correlated to asthma severity. Though how easy and effective it is to obtain sputum samples in the clinic becomes a good question. So.
Do you know if it's increased if you have bronchitis or pneumonia as well?
They didn't look at that. So, the way they kind of use concept studies out is
I was just curious.
Because once bronchitis occurs, then it becomes, how do the pathogen through the pest induces this whole pathway. So the numbers will probably dramatically change.
I hate to do this to you, but sort of followed the first three alarms.
Okay.
Can you explain the biology of this one again, how this plays an inflammatory role,
So this,
and why it correlates?
yeah, and that's where I find it interesting, because while the other ones I could understand is the concept of, in our diseases, we work with the diseases of the outside world injuring the epithelial mucosal barrier, and those alarms being corneas. This is everywhere. This is in our cells. These are the histone controls of our nucleus. And its strong correlation is surprising, without it being muddled by everything else, so, that is a question.
So it isn't intuitive to you either? Why?
It wasn't, Any other questions?
Thank you.
Very good.
Take your boards anytime.
By the way, we forgot to mention, for those for CME credit, the word of the day was molecules. So, the confusing thing to me is those first three molecules have very redundant functions. How do you know which one to target if you're in pharma? Which one looks, if you boil this down to what's a promising direction in clinical medicine? Bill, do you know which is like the leading candidate? Obviously, TSLP, anti-TSLP is being studied.
I thought the IL-33.
IL-33.
It's obviously got some downsides to that one.
If it increases the risk of viral
Full
infection.
circle with the talk today.
What are you showing us there?
Just the alarm is in the middle of atopic dermatitis. 25, 33, and TSLP in there.
Oh, when you talked about IL-17 and all the receptors, I thought that was associated with chronic mucocutaneous candidiasis, but you didn't mention that in any of the risk factors associated with IL-17.
Yeah, there. Aside from that, I mean, it's actually all of these alarmins are associated with an enormous amount of diseases. I mean, like I said, it goes on and on. Corresponds to infection. A whole slew. I mean, there's a whole slew of cancer studies out there associated with these, both in reduction of cancer risks, such as pancreatic cancer, to an increased risk in breast cancer. Then you have a whole slew of all the dermatitis, diseases out there, psoriasis, etc. And it keeps on. I mean, I told Glenn. He asked me when would I have this talk available. I was like, I'm going to work through Thanksgiving on this because I don't even know where to start screening some of this data. So I kind of primarily.
You did a good job. This will need an update. If any of these molecules get closer to clinical relevance as the next biologic risk. Okay, well, thank you all. Anybody know who the speaker is next week? You're the speaker next week. What's your topic?
Epigenetics explained.
Oh, good.
Don't worry, I'll explain it all.
Where are you living?
The one that runs in regular red cat. The cat have you been a regular traditional cat? Subcutaneous full cat cancer. They're given plus or minus that.
When I speak at it,
Do you need anything?
So they're not really using catnip, they're just using
Catnip. That was phase 3 failed.
Oh, really?
Yeah. It didn't meet its
well, I had a really strange reason why on our competition, the placebo results were so out of line.
Yeah, crazy high results.
Yeah, and they were like, well,
That
really
Rex and steady.
they were going to keep their cat. There you go.
Save a phone call.
I know.
Crazy cat owners.
How you doing? Are you fairly
That might be the wrong thing to study.
Unless you're the first person to get a genetically altered cat on the ballot.
No, I'm not.
Then cats would be great.
Have you saw, are you renting or you?
Live a little, huh?
I think that's
Okay, nice to be settled.
yeah.
Good, well, welcome to here. And then I sent, so they changed the Department of Medicine for all the clinical, volunteer clinical faculty appointments.
I can see
That's it.
A little bit of it.
So they've got these gradation of hours that are, we've never had those kind of things now, and it's the first year. So this is one good way of participating in the program. And then you'll be having some volunteers through your clinics at NAC.
Yeah, I mean that's what I right now I'm still like
Yeah.
getting a feel for how it works. So yeah, I would love to.
And then some volunteers that are UW, primarily I guess it'd be UWC, but we'll be starting with Harborview too.
Yeah,
So that would be the other good way to. Yeah, so if that's agreeable, you can make a commitment that I can do that. I think we're okay. Let me see. What are we
Sure.
ready?
Today's Tuesday, Bruce says.
I'm not sure
Rainy
what
day,
time it
but goes
is.
play. So he's the partially wrong.
right to the weekend.
Check with our department days.
Well, that's looking way too far. In fact, this weekend I'm going to
They don't happen until July, but I'm not sure if we've missed.
Because we have the weekend to play, and it's soupy right now.
Oh, I tell you, it's a whole different story.
Career credit change. Yeah, it should
That
be
did
good. Probably
not
second
come
catch up
through.
for
Yeah.
time.
I could hear it.
Okay, it was good to see you again.
Yeah, good seeing you.